students, welcome to a lecture series on geography. The phylogeography is a discipline concerned with various relationships between genealogists, phylogenetics and geography. The word phylogeography was coined by Avis et al. In 1987, and since then the scientific literature has reflected an exploding interest in the topic. In today's episode, we are going to discuss on the topic phylogeography. The topic can be subdivided under the following captions. They are phylogeography, haplotype, comparative phylogeography, phylogeography and biogeography, and applications of phylogeography. Phylogeography. The study of phylogeography grew out of the observation that mitochondrial DNA lineages in natural populations often displayed distinct geographic orientations. In recent years, the field has expanded to include assessments of nuclear as well as cytoplasmic genomes and the relationships among gene trees, population demography, and organism history. Phylogeography has connections to molecular evolutionary genetics, natural history, population biology, paleontology, historical geography, and speciation analysis. The historical biogeography addresses how historical geological, climatic, and ecological conditions influenced the current distribution of species. As part of historical biogeography, researchers had been evaluating the geographical and evolutionary relationships of organisms years before. Two developments during the 1960s and 1970s were particularly important in laying the groundwork for modern phylogeography. The first was the spread of cladistic thought, and the second was the development of plate tectonic theories. The resulting thought was Biogeography biogeography, which explained the origin of new lineages through geological events like the drifting apart of continents or the formation of rivers. When a continuous population is divided by a new river or a new mountain range, two populations are created. Phylogeography, geology, and paleoecology are all important fields that supply information that is integrated into phylogeographic analysis. The phylogeography captures the theoretical and practical richness of the field and also the sense of genuine innovation that phylogeographic perspective have brought to evolutionary studies. Phylogeography is the study of the genetic and geographic structure of populations and species. Phylogeography generally uses genetic information to examine genealogical history and patterning within species and populations. This information is used to infer relationships of biogeographic areas and species histories. The genetic markers utilized are often uniparentally inherited, such as mitochondrial or chloroplast DNA sequences, and as such they track the genealogical history of either the maternal or paternal line. In plants, this is the pollen or seed parent. The genetic discontinuities associated with large geographical gaps between taxa and populations, such as intercontinental breaks, have been relatively well studied. Phylogeography is the process of inferring the phylogenetic relationships among individual sequences and superimposing the resulting haplotype phylogeny over the geographic locations of the samples to reveal population history. They recognized four types of phylogeographic structures. Type 1, deep haplotype trees that are geographically structured. Type 2, deep haplotype trees that are unstructured. Type 3, shallow haplotype trees showing geographic structure. And type 4, shallow unstructured trees. Each type is consistent with a particular history. Geographically structured groups of haplotypes in an area typically result from isolation due to environmental or ecological barriers. In contrast, a type 4 pattern is consistent with recent population expansion and a lack of isolating barriers. Phylogeography can be criticized for being over-dependent on a single gene system, mitochondrial DNA, as a marker of evolutionary descent. The drawbacks, including effects of selection, unplanned increase of pseudogenes, interspecific hybridization, etc., can often be avoided by molecular and numerical analysis and by testing for phylogenetic congruence across nuclear and mitochondrial genes. Coming to haplotype. A haplotype is a combination of alleles at adjacent locations on a chromosomes that are inherited together. A haplotype may consist of one locus, several loci, or an entire chromosome depending on the number of recombination events that have occurred between a given set of loci. Another meaning of the term haplotype is a set of single nucleotide polymorphisms on a single chromosome of a chromosome pair that are associated statistically. It is thought that these associations and the identification of a few alleles of a haplotype sequence can clearly identify all other polymorphic sites in its region. Such information is very valuable for investigating the genetics of common diseases and has been investigated for the human species by the International HapMap project. Many genetic testing companies use the term haplotype to refer to an individual collection of short-term tandem repeat allele mutations with a genetic segment, while using the term haplogroup to refer to the SNP unique event polymorphism mutations, which represents the clade to which a collection of potential haplotypes belong. Coming to haplotype resolution. An organism's genotype may not define its haplotype individually. For individuals who are homozygous at one or both loci, the haplotypes are clear. For individuals heterozygous at both loci, the gametic phase is ambiguous. The only plain method of resolving phase ambiguity is by sequencing. However, it is possible to estimate the probability of a particular haplotype when phase is ambiguous using a sample of individuals. Given the genotypes for a number of individuals, the haplotypes can be inferred by haplotype resolution or haplotype phasing techniques. These methods work by applying the observation that certain haplotypes are common in certain genomic regions. Therefore, given a set of possible haplotype resolutions, those that use fewer different haplotypes overall. The specificities of these methods vary. Some are based on combinatorial approaches, whereas others use likelihood functions based on different models and assumptions such as Hardy-Weinberg principles, the coalescent theory model, or perfect phylogeny. The parameters in these models are then estimated using algorithms such as expectation maximization algorithm, Markov chain Monte Carlo, or hidden Markov models. Why DNA haplotypes from genealogical DNA tests? Unlike other chromosomes, Y chromosomes do not come in pairs. Every human male has only one copy of that chromosome. This means that there is any chance variation of which copy is inherited and also not any shuffling between copies of recombination. So, unlike autosomal haplotypes, there is effectively not any randomization of the Y chromosome haplotype between generations. A human male should largely share the same Y chromosome as his father, give or take a few mutations. Thus, Y chromosomes tend to pass largely intact from father to son with a small but accumulating numbers of mutations that can serve to differentiate male lineages. Y chromosome and mitochondrial DNA haplogroups have different haplogroup designations. Haplogroups pertain to deep ancestral origins dating back thousands of years. Haplogroups. Haplogroups pertain to deep ancestral origins dating back thousands of years. In human genetics, the haplogroups most commonly studied are Y chromosome haplogroups and mitochondrial haplogroups, both of which can be used to define genetic populations. In human genetics, the haplogroups most commonly studied are Y chromosome haplogroups and mitochondrial DNA haplogroups. According to research, Y DNA is passed exclusively along the patrilineal line from father to son, while mitochondrial DNA is passed down the matrilineal line from mother to offspring of both sexes. It is believed that neither recombinase and thus Y DNA and mitochondrial DNA change only by chance mutation at each generation with no intermixture between parents' genetic material. A new haplogroup is born when a very specific new mutation occurs. All descendants will carry that mutation. That mutation defines that haplogroups. So, if a new haplogroup is born today, we would not know it was a haplogroup until hundreds or thousands of years later when we see that lots of people have this same mutation from a single individual. As you might imagine, many haplogroups over the ages have died out, but some have been very successful as evidenced by the fact that we are all here today. Coming to comparative phylogeography. Comparison of phylogeographic patterns among broadly sympatric species shows whether species have responded in parallel to recent isolating events. The first step is to find out if reciprocally monophyletic groups exist within a sample taxa in a widespread community. The next step is to assess if the groups are sister groups and whether the reciprocally monophyletic groups are geographically concordant. If so, one can determine how long the groups have been isolated either in a relative sense or by raising a molecular timer. Next, assuming there is a congruence indicating a historical barrier to at least some taxa, one examines reasons why other species in the current fauna do not show reciprocal monophyly across the same putative barrier. Either species were widespread and simply did not respond to the barriers that caused differentiation in other species, or they recently expanded their range across the barriers. The species with genetic signature of recent expansion might be relatively recent arrivals in the current community. Alternatively, they might have been widespread and gone through an obstacle followed by expansion. The field of comparative phylogeography seeks to explain the mechanism responsible for the phylogenetic relationships and distribution of different species. For example, comparisons across multiple taxa can clarify the histories of biogeographical regions. The phylogeography also gives an important historical perspective on community composition. History is relevant to regional and local diversity in two ways. One way is the size and makeup of the regional species pool results from the balance of speciation and extinction. Second way is at a local level community composition is influenced by the interaction between local extinction of species population and recolonization. The comparative phylogeography reveals the historical stability of the current community. If the current species pool was historically coherent, widespread and bisected by an isolating barrier, a prevalence of geographically similar reciprocally monophyletic patterns should be recovered. The comparative phylogeography analysis can contribute to broader studies of ecology and evolution in a number of ways. First, phylogeographic analysis can identify historically and evolutionarily independent regions that can be considered as natural replicates amongst which generalizations about specific processes can be tested statistically. Second, phylogeography can provide an evolutionary and geographical context for the species comprising ecological communities, thus permitting determination of historical and spatial influences on patterns of species richness, and understanding of historical responses to changes in the landscape, and the identification of evolutionarily isolated areas can inform conservation strategies. Comparative phylogeography describes the evolution of landscapes and permits analysis of the effects of history and geography on organismal community structure at both local and regional levels. The knowledge concerning the different ages of biota and their areas of extent and the different rates of species origin and extinction will enhance the understanding of processes responsible for the origin and maintenance of communities and make some contribution to conserve biodiversity. Next, we shall move on to comparison between biogeography and phylogeography. The diversification at all taxonomic levels has a special and temporal component. Historical biogeography and phylogeography represent analysis at different points on the range of evolutionary divergence, differing levels of history. The historical biogeography deals with phylogenetic patterns among species and higher lineages attributable to relatively ancient events in Earth history. The phylogeography deals with the origins and relationships of taxa at the lowest level of taxonomic organization. There is a distinction between phylogeography and biogeography. Phylogeography can involve historical conclusion even if phylogenetically structured trees are lacking, whereas biogeography typically requires resolved trees. However, the distinction between biogeography and phylogeography blurs when one discovers that a species includes multiple equally monophyletic groups related in a hierarchical way. Not surprisingly, there are parallels in the theory and methods employed. The phylogeography and biogeography are established on phylogenetic analysis. Both are based on analysis of single taxa, that is, clades in biogeography and species in phylogeography. But they are most powerful when comparisons of multiple lineages and species are involved. The historical biogeography seeks to find harmonious phylogenetic patterns in multiple clades found in a common region. Similarity is interpreted to mean that a widespread ancestral biota was fragmented by a series of successive biogeographic events. The comparative phylogeography seeks to find concordant splits within simultaneous co-distributed species. The degree of phylogeographic concordance is a measure of the historical stability of the current species group. Both historical biogeography and comparative phylogeography reveal the historical stability of communities at different temporal scales. Similar phylogenetic or phylogeographic patterns provide relatively straightforward inferences. Biogeography and comparative phylogeography differ in their potential to explain different patterns owing to the disparate timescales, dispersal, extinctions, and overlapping events in Earth history can unclear biogeographic history. The reasons why clades have dissimilar phylogenetic histories are difficult to ascertain because of the age of the events. The traces grows older with time. In contrast, comparative phylogeography deals with relatively recent events, and genetic signatures often reveal species history irrespective of the geographic pattern of haplotype relationships. Thus, it is possible to read the history of each species whether the haplotype tree is structured or not. Now, coming to applications of phylogeography. The power of comparative phylogeography derives from analysis of many co-distributed species. By tabulating the distribution of phylogeographic types, one gains an understanding of a fauna's recent historical stability, and by comparing distributions among communities, one can infer which current communities have changed the least in species composition over the recent past. Thus, comparative phylogeography offers a means to diagnose the history of biotic stability. Phylogeography, the geographic distribution of genetic lineages, to clarify biogeography and apply the results to issues in conservation biology. Fundamental to these activities is understanding the sources and distribution of variation, that is, the seeds of speciation. Thus, phylogeography has a central role in evolutionary biology at the boundary of microevolution and macroevolution. The phylogeography also has proven an immensely useful approach to questions of species conservation and resource management. The phylogeography is a rich, integrative discipline that demands consideration of, among others, paleontology, historical geography, molecular evolution, and ecology. Its application and products are of interest to ecologists, evolutionary biologists, conservationists, and managers of coastal resource alike. Phylogeography seeks to test the congruence between the evolutionary, demographic, and distributional histories of taxa against the particular geological and ecological setting of a region, and to determine the chronology of evolutionary diversification. The field of phylogeography developed with the increased availability of DNA sequence haplotype data. These data were analytically suitable for using phylogenetic analytical techniques, but were variable enough to address intraspecific questions. These questions include ones of taxonomy and or demographic limits, migration patterns, and how abiotic environmental changes influence biotic communities. Although early phylogeography studies relied on qualitative analysis of the phylogenetic relationship of individuals, the field has become increasingly statistical in nature and in many ways has returned to the quantitative models developed by population genetics. The focus of the phylogeographic approach has been on reconstructing the geographic history of a species or group of species. The influence of phylogeography is spreading throughout biology. Among other examples, phylogeographic techniques have enabled us to infer the origin of mice, modern humans, and man's best friend, the domesticated dog. The phylogeographic analyses also enable public health officials to understand the origin and spread of emerging infectious diseases. In spite of this success, disagreement and confusion persist regarding the most effective ways to learn about phylogeographic processes from geospatially identified molecular sequence data. Phylogeographic methods facilitate inference of the geographical history of genetic lineages. Recent examples explore human migration and the origins of viral pandemics. There is long-lasting disagreement over the use and validity of certain phylogeographic inference methodologies. The phylogeography has also proven to be useful in understanding the origin and dispersal patterns of Homo sapiens. Based on primary observations of skeletal remains of ancient human remains and estimations of their age, anthropologists proposed two competing hypotheses about human origins. However, in light of these recent data from the 1000 Genome Project, databases sampling thousands of individuals globally and samples taken from two non-Homo sapiens hominins, the picture of human evolutionary has become more resort. Finally, coming to conclusion, phylogeography is a rapidly growing discipline that aims at studying the principal and historical processes governing the geographical distributions of genealogical lineages. In contrast to classical population genetics, which is based on allele frequency distributions, phylogeographic procedures are supposed to separate population structure from population history. Thank you.